Good morning, everyone. My name is Ben Kleinstiver. I run a research lab at Mass Gen Hospital and Harvard Medical School. It's my real pleasure to moderate this session on gene editing and precision medicine this morning. I think it'd be an understatement to say that it's been a really phenomenal year for the gene editing field. Really tremendous successes both in academia and in industry in terms of developing gene editing drugs for patients, including, most notably, to be shown on a slide soon, 3 individuals that have been some of the first for our field to receive gene editing treatments. One, Alyssa Tapley, she had refractory T-cell leukemia and is now in remission. Second was baby KJ Muldoon, who had an inborn error of metabolism, a urea cycle disorder, and now has been treated and able to live a relatively normal life and consume protein. And third, Tracy Atterbury, who had an inborn error of immunity and received one of the first prime-edited medicines, and again, also now living a relatively normal life. So these are just 3 of hundreds of patients that have been treated in clinical trials, dozens of clinical trials for gene editing medicines, all showing very promising signs of being beneficial to patients. And yet, despite this promise, I think there have been some challenges in the field with companies really contracting their pipelines and focusing on very specific programs. So on this panel, I think we'll reflect on some of the real successes in the field over the past year and some of the challenges and opportunities moving forward. So to start, I'll just invite each of the panelists to introduce themselves briefly and maybe reflect on something that happened in the past year that has really been sort of emblematic to them of the success in the gene editing field. So maybe start with Omar.
Great. Hi, I'm Omar. I'm an assistant professor at Harvard Medical School, and my lab works on really building programmable tools for biology, including new ways to do gene editing. And yeah, just ditto to what Ben said. It's been an incredible year to actually see baby KJ get treated. I think one of the challenges with genomic medicines, especially gene editing tools, is there's a very long tail of mutations, right? Even though there's 7,000 genetic diseases, there's technically millions of potential pathogenic genetic variants. And so the idea of treating all of those with the current systems in place for drug development is very daunting. And so the idea that you can go from sort of concept to deployed drug in a person within, I don't know, like 6 months or so is incredible. And thinking through how to sort of reproduce that at scale, I think, is a very exciting prospect. So excited to be here today.
Thanks, Omar. Steve Favaloro. I'm the chairman and CEO at Genozyne. I have the great fortune of leading the team of about 300 colleagues passionate about making medicine. We're on the CMC and manufacturing side. Spent about a decade in the advanced therapy space, almost all on the manufacturing side for editing and for gene therapies. What's so exciting to me about this year and just generally about the space is just the advances in manufacturing and CMC that enable these medicines to go to patient populations, to very small patient populations, even to N of 1. And it leaves me very, very optimistic about the space and the future of gene editing and gene therapy.
Hi, I'm David Liu. I'm a professor at the Broad Institute and Harvard and HHMI. And our lab works on gene editing, among other topics. And our lab developed base editing and prime editing, some of the technologies that were used to treat the patients in Ben's slide. It is a remarkable state right now for the field of gene editing. We saw the kinds of landmark medical milestones that Ben just summarized beautifully. Just in the past year, there have been several. First, correction of a pathogenic mutation in a person happened in the last year with the work of scientists at Beam Therapeutics correcting the most common cause of alpha-1 antitrypsin disorder, which is fairly common genetic disease as rare genetic diseases go. The first bespoke gene editing treatment in baby KJ Muldoon's base editor to correct one of baby KJ's pathogenic mutations in a gene called CPS1. First clinical readout of prime edited medicine to treat a disease called chronic granulomatous disease, treating, among other patients, Tracy Atterbury. So the capabilities of the field have never been more rich with opportunity, more exciting, and I think the field has never felt more obligated to try to connect the science with the people. And in some cases, for example, LNP-mediated mRNA delivery to the liver, thanks to the hard work of not just the scientists, but also the people working on the manufacturing, regulatory agencies, there are now, I think, robust opportunities to treat patients with many genetic diseases that affect many, many people. As Omar mentioned, I think there's also a real need to continue to use the science to innovate ways that can bring the promise, the value of gene editing to larger and larger numbers of people, recognizing that we need increasingly allele agnostic ways, mutation agnostic ways of treating patients, and even perhaps some disease agnostic ways of using gene editing to provide one-time treatments for different diseases that share common information flow defects in the cell. And I think we'll see more of that coming in the near future.
Hi, I'm Trevor. I'm co-founder and CEO of a company called Mammoth Biosciences. No, we're not the one resurrecting mammoths. Spun out of Jennifer Doudna's lab 8 years ago, and what we're really excited about is kind of achieving the holy grail of genetic medicine, which is how can you actually make any kind of edit in any cell. So we have a lead program in the liver. We have a lot of work that we're doing in muscle and CNS across different types of editing modalities. And I would say that you have to get more creative as people keep answering the question, but I think one thing that's been, I think, a little bit under the radar for the space is that the FDA is actually moving forward with a lot of these like platform designations for the technologies, and I think that is one of the key unlocks for a technology like gene editing, where a lot of the promise is that you can use a lot of the same building blocks to go after many, many different diseases, and I think that that could be very transformative over the next 5 or 10 years.
I'm Laura Sepler-Encino, and I'm a drug developer. I had the opportunity of being part of 2 firsts. The first one was during my time at Vertex working with CRISPR Therapeutics. We developed Casgevy, the very first CRISPR approval for sickle cell disease and beta thalassemia. And then I joined Intellia Therapeutics as the CSO, and there we were able to demonstrate that CRISPR in vivo works in humans. For 2 programs, demonstrating the power of a platform approach using an LNP for delivery to liver, so we can tackle any diseases, genetic diseases that are driven by genes expressed in the liver. You know, with the work that we have done, you know, these 2 companies, right, we have paved the way for, you know, these new advancements. Right, these new editing enzymes, these new ways of approaching disease with truly validated delivery modes for hematopoietic stem cells and liver. I think that what we need to do now is, you know, I think we have plenty of tools, and there are more tools coming up for editing, you know, precise editing. Where the gap still remains is on delivery. And this is, you know, where as a field we need to, you know, get new delivery modalities that truly break up, you know, opens up the disease space. You know, and then continue our partnership with regulatory agencies to, you know, as we get more and more human safety data and durability data, build that, you know, sense of safety and utility of these platforms so they can be more broadly applicable.
Wonderful. So I think that really nicely sort of lays the groundwork and framework for this session. A lot of really exciting news in the past year and opportunities moving forward. So I think next we're going to take a deeper dive on this treatment for baby KJ. So if you can put that slide back up one more time, please, that would be wonderful. This is an individual who was diagnosed a few days after birth with a severe metabolic disease. And what ensued after that at the University of Pennsylvania and at CHOP with Kiran Musunuru and Becca Ahrens-Nicholas leading the clinical efforts was nothing short of simply a scientific whirlwind and a real impressive feat. So within 7 months, a customized gene editing treatment was developed specifically for baby KJ's mutation, dosed into the patient, actually receiving 3 doses over the course of a couple weeks. And now showing signs of really tremendous benefit. KJ can now eat a roughly normal protein diet and has gone home for the first time with his family. So I think really this is a remarkable success. To go from diagnosis to treatment in about half a year is a real testament to where the field is now, leaning on the shoulders of giants like Intellia and others who really pioneered the development of in vivo gene editing. So maybe, David, you could talk a little bit about the technologies that underlied the base editing approach and how those were developed in your lab and how really this has been impressive to achieve this feat in such a short period of time.
Yeah, and to me, the most important learning from the baby KJ story is the fact that no new science had to be invented, which is the only reason how it could possibly have been done in less than 7 months, something that would normally take, as I like to say, roughly 7 years. This is in part due, for sure, to the extraordinary leadership and organizational skills and goodwill of all the participating labs, of Rebecca and Kieran, most notably. But it's a remarkable testament to how robust some aspects of the gene editing enterprise for human therapeutics currently are. And while I completely agree with Laura's statement that we need to advance the science in many other directions to maximize the ability of patients to benefit from these technologies, delivery being one of the most obvious, it is a remarkable testament to how robust the science, the manufacturing, the regulatory state are that we can do something like what was just accomplished. So to answer the other part of your question, Ben, base editors, for those who don't know, use the beautiful targeting mechanism of programmable DNA binding proteins like disabled CRISPR scissors or tail proteins or zinc finger proteins to engage a DNA sequence of your choosing. But instead of cutting the DNA to disrupt a gene, base editors perform chemistry on an individual DNA base, converting it from one base, like A, into a different base, like G, or technically a base that pairs like G, behaves like G. So as you might imagine, the most common kinds of mutations that cause genetic disease turn out to be single letter changes, just because they're the most frequent that spontaneously happen during the course of a day or during the course of the DNA replication or the formation of a child. So base editors can correct single letter mutations that cause many, not all, but many genetic diseases. About 1/3 of the 200,000 or so known pathogenic mutations can be, I'd say, efficiently corrected using a base editor. So that's the core technology that was used to correct one of baby KJ's 2 mutations in a gene called CPS1. KJ inherited 1 mutation from dad, 1 mutation from mom. Mom and dad are both fine because they have 1 good copy from each of their grandparents, but baby KJ, unfortunately, just as Mendelian genetics would predict, was the one of the 4 kids who had both mutations, and that resulted in this severe disease that was under extreme time pressure to correct. And so baby KJ's father's mutation was the one that this big team corrected using an adenine base editor. That's
great, David. So Omar, you're the director of the gene editing core at the Gene and Cell Therapy Institute. Maybe you can talk about how, in the KJ therapy, this was an essential collaboration between academia and industry that could not have been accomplished in the absence of each other. So how do you think about the role of academia and industry working together moving forward for these types of therapies?
Yeah, sure. That's a great question. I mean, I think David just gave an incredible sort of explanation of, the technology that goes into really being able to make atomic level changes to correct these mutations in people, right? But to take that technology and actually get it to work as a therapy, there's actually a lot of complexity to make like an actual drug product, right? So like a gene editing modality is not just, this single thing that you can just inject into someone and it'll work, right? You have to figure out how to express the protein component components. You have to figure out how to express this piece of RNA called the guide RNA that actually targets the protein to the right place. And then you have to figure out how to package it all together in a way that it can actually get to the cell type you want. And David also hinted at this, right? Like delivery technologies need to be better, and there's this thing called lipid nanoparticles, which I'm sure everyone in this room probably has been injected with because of COVID vaccines. And so that same technology is what we leverage here. And it turns out a lot of these things are quite difficult to actually design and make and scale up for human level sort of scale. mRNAs are notoriously difficult to design correctly and produce. Guide RNAs, took many years for us as a field to even figure out how to get them to be stable inside the cells and not get degraded immediately, and there's a lot of chemistry that goes into that. And then lipid nanoparticles themselves can be, they can be toxic. It's so often that we design a lipid formulation that kills mice. And so you have to be really thoughtful and careful with how you design them to get them to the right cells. And so that turns out to be a really big optimization problem that companies and industry are really good at, right? And so that's, I think, where this multi sort of academic and industry collaboration really came into play. Like Danaher, for example, has really accumulated all this expertise around all of these components, right? From guide RNA chemical synthesis to mRNA design and production to I think even lipid nanoparticles. And so that ability, and because of I would say actually because of our experience of the COVID vaccine and scaling up mRNA nanoparticles at scale across the world for that, I think a lot of that went into enabling us to move quickly with the KJ drug. And when we think about the GCTI and the gene editing core even at Mass General Brigham, we want to think about how to bring that to our ecosystem, right, in Boston, right? If any clinical research lab has a mutation or disease that they think could be treated, it'd be really amazing to have the infrastructure to go right from concept to picking the right modality. Now there's, I don't know, David, like 7 or 9 generations of base editors and many generations of prime editors. So picking the right one, figuring out how to produce it, and making a pipeline into patients would be really the vision going forward.
That's great. So Steve, I guess coming from the manufacturing world, how impressive was it to you to see all of this happen in such a short period of time?
Yeah, it's so impressive, remarkable. That the underlying science is astonishing, right? And then we need to figure out how do we make these at scale. And the difficult part, when we say scale, that could be an N of 1, an N of 10, ultra rare, or it could be a 2,000 patient population. So those are different things. And we need to do it safely and under GMP. So the advances in manufacturing have been profound the last decade, and what excites me is the learnings from the last decade of making these commercially, what that's going to teach us for the next 10, 20, 30 years of being able to make these things, either from an N of 1 to a larger patient population. And I think that's been both from an equipment side and a technology side, but also just an underlying learnings of the process development, and most importantly, the analytics, and how do you test that something is safe and releasable into a commercial product after that. We're going the route within gene and cell, gene editing, of getting to a place where these become more standard manufacturing processes, which will enable the whole field, and get regulatory agencies more comfortable with the approvals and working in tandem with the science to make sure that these can get to patients.
You guys are doing an amazing job of teeing up the next questions here. So, Laura, maybe you can talk a bit about the speed of the regulatory interactions and how maybe this has been possible just given that the FDA was familiar with these types of gene editing drugs from pioneers like Verve and Intellia and others.
Well, so it's exactly that, right? So the fact that there was a precedent, and a very robust precedent, not just from one company, Intellia, but from others, right? Demonstrating that you can make a lipid nanoparticle, you can select a guide RNA with the qualities, on target and, characterization of potential off targets, de-risking, all the preclinical data demonstrating, safety. And that blueprint could be, applied to these bespoke therapies. So, if that hadn't been in place, right, it would be a different story, right? Because, there are many things that are uncertain. But this is what, as a field, we can help each other and, really try to establish a knowledge base that can be more applicable, right? Where certain things you have regulatory interactions that build confidence on the approach and facilitates, whether it's bespoke therapies, but also the extension of that to more prevalent diseases.
Yeah, that's great. So then, Trevor, maybe you can talk a little bit about the aspect of safety as we think about developing these gene editing drugs, especially for a therapy like this where you have a limited amount of time to intervene with a child. How should academics and companies think about the depth of safety studies versus the balance of time in these types of situations?
Yeah, I think for me, the most interesting thing on this is that I think now there is quite a large body of work from multiple companies, multiple academic labs looking at safety in various different directions, whether that's of the delivery vehicle, of off-target, et cetera. And I think to me now, there's not really much debate for like really high unmet need diseases, like whether that's a child or an adult. I think we actually have like quite a good body of work. We know kind of where we stand on that side. I think the more interesting thing going forward that also connects back to maybe how you started the conversation in terms of it's never been more exciting in terms of the results we're seeing in gene editing, but if I'm being honest, it's never been more depressing in terms of like the level of investment and excitement around it from like an economic side. And I think part of bridging that gap is related to safety, which is how do you go from rare to more common disease? And if we look at a space that is kind of the opposite of that, it's having a renaissance right now. It's like siRNA, like think of genetic medicine. There are spaces that are seeing massive investment. And I think that's in large part because those technologies have made the jump now where you're not talking about 10 patients or 100 patients or 1,000 patients. You're talking about 10,000 patients, 100,000 patients, maybe even 1 million patients. And I think that's going to be the big question on my mind for gene editing is when do we get to that point? Can you like imagine, so speaking of last year, the acquisition of Verve, right? So you have a major pharma company making a real bet on, I think, an appropriately ambitious vision of how do you actually reduce cardiovascular disease. Like, can you imagine how many lives you could save if you could essentially genetically inoculate people against various forms of cardiovascular problems? And I think that, at its core, is largely a safety question, right? Because we know it functionally works. It's just a question of when you're dosing hundreds of thousands to millions of people, is the risk-benefit calculation correct? And I think what's exciting is through pioneering work from companies like Intellia, where now there's hundreds of patients dosed, and we followed them for many years. I think there's always a new kind of barrier where you can say, oh, look at them 5 years out, 10 years out, 15 years out, and that's appropriate, right? But I think what's been really exciting is that even with the quote-unquote first-gen technology, and I say this as a company that has all sorts of new-gen technologies, so I have every reason to be like, oh, no, you need the version blue of CRISPR or whatever. It turns out it's actually incredibly safe. Like, incredibly safe. I think we take that for granted. So I think that's what I'm excited about on that side is you'll always have kind of a new goalpost in terms of, let's look at this other safety parameter, and that's appropriate. But I think it's been amazing how genetic editing has really shown to be honestly safer than I think most people would have predicted at the beginning.
Yeah, I guess maybe, like you say, with the balance of time, we can better understand the real risk, of 5-10 years out of these treatments. So maybe opening up to the panel, anyone that wants to dive in on this, what is the opportunity to do this in less than 7 months? Can we do this faster? And how do we use this blueprint of baby KJ to get this into more patients with rare diseases?
Yeah, I think we can definitely do it faster, even though 7 months is remarkably fast. A lot of that 7 months was spent creating and doing experiments on animal models. In some cases, one might argue without too much learnings from the results. It was really designed to conform to anticipated regulatory requirements. Of course, FDA and other regulatory bodies, including many outside of the US, are very aware that they are one of the main bottlenecks in this whole enterprise, not just from the time needed to develop these drugs, but the programs that a company will take on are all gated by whether they believe they can at least recover the costs needed to bring these drug candidates to clinical trials and eventually into products. And much of that cost is determined by what are the regulatory requirements needed to meet all of the standards in the countries that you wish to make the drug available in. Those are important, of course. We want to make sure that, above all else, we do what we can to practically minimize harm, the risk of harm to patients. But if you look at the history of medicine, there are learnings from past platformization of drugs and of processes that I think gene editing is now in the process of adopting. You know, a cancer vaccine is not approved on the basis of an individual composition of matter. Otherwise, everybody would have to go through a different review process because everybody's cancer vaccine is different. When you get a blood transfusion, one might argue that the bag of blood from one donor and the bag of blood from a different donor in many ways are, on average, more different than a base editor programmed to fix baby KJ's mutation versus a base editor programmed to fix a different mutation in a different gene in the liver, from a composition of matter point of view and perhaps even from a risk point of view. But bags of blood are not reviewed and approved on an individual bag basis. The process is the product that's approved. So I think we'll see a lot of movement, as FDA is already contemplating, moving in ways that make the regulation more of gene editing, programmable gene editing medicines, more like approving a process rather than approving a product. So that, I think, will have a huge impact as regulators begin to get comfortable, whether that's in this country first or in other countries first, of really taking advantage of the high degree of similarity between baby KJ's base editor and the base editor used to correct the first alpha-1 antitrypsin deficiency mutation in a different patient, both delivered by mRNA delivery using an LNP into the liver. So that's where I think we'll start to see real meaningful steps towards the future that Trevor just articulated.
Yeah, maybe to add to that, right? So this platformization, right? So if you have modules that are, you know, talking about how to accelerate, right? So modules that are fully validated that you can pre-mate, right? So, and for which you could have master drug files that you can use for regulatory submission, you know, that can accelerate, right? So if you have a set of editing enzymes that you already know that they're useful and you can pre-make the mRNA for them, you know, and you have the composition optimized of your LNP and you understand what's the preclinical path, right? So that could certainly accelerate time to dosing, from diagnosis to dosing.
Yeah, that's great. And I think, you know, to dovetail off what David said, I think the editor enzyme for baby KJ was essentially locked in a matter of weeks. That decision had already been made. And the remainder of those 5 or 6 months were doing the safety studies, which I think are obviously important, as you note, but as we learn from this and other programs over and over again, maybe the necessity of each of those components may be less important if the time becomes a more critical factor in getting this earlier into patients. So then how do we think about getting this into more patients? Is this a technology challenge? Can we develop other editing approaches that allow us to think about, you know, one-time treatments for all patients with a mutation in a single gene? Omar, your lab's done some work on these types of approaches.
Sure. Yeah, I mean, I think there's at least 2 limitations. I'm sure the panel could think of more, but two I'll talk about. One, I think, is technology limitations and specifically delivery being one of the main challenges. And the second, I think, being how do you figure out the economics of scaling this up to a lot of people? Because it's still, even if you know, figured out the platformization and, you know, how to make it faster, it still costs a lot, right? And, you know, rare disease is quite rare. In the long tail, you know, there's not a lot of patients, and so it becomes still a bit of a commercial challenge to figure that out. So on the first part, I actually think, you know, gene editing technologies, there's still some work to do. But for the most part, they're getting to the point where they're pretty mature and they can work for most mutation types. And, you know, there's caveats like, can we get the right efficiency in the right cell types? You know, sometimes certain cell types are harder to edit than others. But for the most part, what's limiting us to going after a lot of these diseases is like just being able to reach the right tissues, right? You know, it's very easy to deliver to the liver in organisms just because the blood gets filtered there and nanoparticles are easily taken up. But other places like brain, kidney, heart, muscle, you know, are really challenging. And that's a big reason why a lot of diseases you hear about, at least in vivo, that are edited are liver diseases like baby KJ. And so if we could really solve the delivery challenge, that would open up treatment for a lot of people. And then on the financial side, one way to solve that, as multiple folks have said on the panel, platformization helps. Another technology-driven way to solve that is something that my group works on, David's group also works on, which is this concept of large sequence insertion or replacement. And the concept is, instead of targeting one mutation at a time, can you target many mutations at once, like replace a whole exon, replace a whole gene. And there, all the work that goes into developing your product is thus applicable to way more people. For example, all the people that have a mutation in a given disease. And for example, cystic fibrosis, right, is like thousands of mutations. And most diseases have dozens, if not hundreds of mutations. And so that could solve a big piece of economic puzzle as well. And then I think there's other sort of structural parts. Like if you could figure out centers of excellence or whatever for doing this type of correction in academic hospital centers, I think there's other ways, creative ways you could think about funding it, whether it's through government or insurance or whatnot. But that's another, I think, critical piece that has to be addressed.
Yeah, I think those new structures are coming. Our government, HHS and FDA, are very aware of the points Omar just articulated. And I think they also feel the need and the mismatch currently between investment and need. So Global Genes recently showed me statistics that I found illuminating. There are 39 million people in this world that live with HIV/AIDS. If you look at global investment in HIV/AIDS research, it breaks down to $500 per patient for those 39 million patients. There are about 50 million patients living with cancer right now. $10,000 per patient is the global investment in cancer. There are 400 million people living with a rare genetic disease now. So in aggregate, not so rare. Total global investment per person, per patient for rare genetic disease is $3. So those are our statistics from Global Genes, which tracks these things. And that creates a mismatch, which creates both urgent need and an opportunity for the brave investors and companies out there who want to have more of an impact on these 400 million people, many of whom suffer from serious diseases like baby KJs, that for them it's a life-changing, family life-defining treatment that they're seeking. So not so rare in aggregate, and I think that ultimately will help drive help guide, I hope, a lot of the advances in this space.
Yeah, definitely. I think a lot of opportunities moving forward. So I had so much planned to ask you guys and discuss, but we're running out of time. So maybe let's look over the horizon a little bit and just maybe in a couple sentences each talk about the things you're excited in the next 2, 5, 10 years. What do you think will be the big leaps for the field, and what are you most excited about moving forward?
Well, I'll start. Look, I'm excited about what we've done thus far and what's coming. So for me personally, I think that we're going to have the first approval for in vivo genome editing for hereditary angioedema and transthyretin amyloidosis, and those are going to be transformational. Can we begin to talk about cures? I think that cure means, implies a finality, and it's a bit early to talk about longer term effects. But, all the data that, I'm seeing, particularly for hereditary angioedema, where people have attacks, they get dosed, and those attacks go away, give me confidence that, this could be, a potential in the future. Then is, as you're beginning to build that human safety database and that comfort that, we know what we're doing at drug developers and, as a tool developers, right, that we can do this with quality and safety. Is the opportunity to go now from the bespoke individualized therapies to, the targeting risk factors, right, for cardiovascular disease, right, and that spectrum of diseases and spectrum of personal risk-taking that you may want to take. And I believe that I was involved in RNAi, 20 years ago, And I remember all the discussions about RNAi being unspecific and it's going to be toxic, and now there are millions of people taking it. The same thing is going to happen with genome editing technologies.
Yeah, and I totally agree. I think one of the most exciting things is that we are seeing this transition from ex vivo to in vivo, and I think in large part to work Intellia has done. I think we'll see the first approvals. I'd be even more ambitious. Like, I don't see why anyone has to live with a liver genetic disease like 5 to 10 years from now. I think that is an incredible statement to be able to make, but I think you can genuinely say it with a straight face. I think what I'm most excited about beyond that, though, is how do we move beyond the liver? We have all these amazing editing technologies. There's all these different delivery methods out there. I don't think you necessarily have to invent new ones, but when I think about genetic medicine, I think about like Alzheimer's, Parkinson's, ALS. Like, these are the diseases that I think the average individual on the street would think about. Huntington's. These are diseases where we've even mapped the genetic cause for half a century for some of them at this point, yet still today people die from them. So I think genetic medicine will have truly arrived when like the last patient dies of Huntington's disease. And I think that for the first time, you can actually kind of see a path towards these different areas. So I think really having the same level of confidence in our engineering and delivery that we have for liver, for like muscle, CNS, heart, all these other areas, I think that is going to be transformative over the next 5 to 10 years. And I think as part of that as well, moving to more common disease. So as we show that we're extremely safe in the liver and we can move to more risk factors for things like cardiovascular, how do we do the same for every other tissue in the body?
Yeah, I share many of the same sentiments from Laura and Trevor. I think in the shortish term, so 5 to 10 years, we'll see some key approvals. Laura mentioned a couple. In the base editing, prime editing space, there are 23 clinical trials ongoing now. 8 have already read out. I think in the next 5-ish years, we'll see the first base editing and prime editing approvals, which will open up, serve as a precedent to treat many diseases that require gene correction or more precise conversion of one sequence to another sequence in order to benefit patients. And then in the longer term, I think we'll start to see both newer tissues open up to gene editing, which don't necessarily require the invention of new delivery modalities. There are among those 23 base editing and prime editing clinical trials, 0 currently use AAV. That's obviously, we would be able to treat diseases in many more tissues if we could use AAV, which is the only way we know of to clinically deliver into a variety of important tissues in your body right now. Hopefully that will change over time, but I think we'll start to see some of the first clinical trials and hopefully positive clinical readouts using AAV to deliver these gene editing modalities. And then I think also in the sort of long-term bucket, we'll start to see some of these mutation agnostic and hopefully some disease agnostic approaches reach the clinic and benefit patients, where we're not just correcting an individual's mutation like baby KJ's, we are instead editing the gene in a way that, as Omar articulated, could benefit any patient with a mutation in that gene, and even more broadly, making one-time edits elsewhere in the body, elsewhere in the genome, that might treat many patients with different diseases, but that all suffer from similar information flow defects, like premature stop codons. We are showing in our lab can be treated both in cells and in animals by using prime editing to rewrite a dispensable redundant tRNA that you don't need in your body, converting it permanently into a very efficient suppressor tRNA, but still under endogenous gene control. So I think it'll be an important 5, 10, 15 years for the field that will help bring the science to many, many more patients than the ones that have already been treated, even though those represent medical milestones by themselves, and fortunately there are no examples I'm aware of where gene editing has not benefited patients, and fortunately no examples where from the clinical trials underway so far where where the editing has harmed patients, as opposed to side effects from other things that are required in the trial.
Thanks, David. I'm really excited for the next decade in gene editing, gene therapy. The last decade is on the CMC side, especially the science side, but the CMC side has shown us so much on how to make these medicines. Thinking back to the first approval in 2015 with Kymriah for an ex vivo cell therapy to hopefully many approvals a year as we look forward. The technology has drastically improved, both from an input perspective, thinking through to the future on maybe using synthetic plasmids as an input, to the actual tools that we're using to manufacture these therapies that are safe and efficacious. And that means we need to get them to patients, and that's something we take very seriously as a manufacturer and as someone who enables the space to get these medicines to the patients that need them.
Great. I guess we haven't talked about AI, so let's fix that. So I think for gene editing, I think there's a few different ways AI could actually really help. Maybe at the top of the funnel, right? Like, I think, we assume like, someone, a child presents with a disease and it's a clear-cut case, like, oh, this is the mutation they have. And in reality, I think it's actually, can be quite difficult. There's a lot of, just imagine, your child has symptoms and you show up and there's a lot of different diseases that those symptoms could fall in, and you do whole genome sequencing and then you see all these mutations, and it's actually quite difficult to be a lot of times to say like this is the mutation for sure causing that disease. And so I think there's a big role for AI to play in helping to really look at complex patterns in your genetics to try to map the mutations that you might want to correct with gene editing. And for just as an example, Catherine Brownstein at Children's has been doing a lot of great work on clinical pediatric genetics for this purpose with actually OpenAI. So I think that's one exciting area. I think another exciting area is just using AI. We talked a lot about speed today, right? How do we quickly go from someone's mutation to the drug product, and part of that speed is making sure that when you design your mRNA or your guide RNA or your protein editor, that it's not messed up along the way. And when I say messed up, I mean like it's too inefficient for this particular disease, and that slows you down, right? And so AI can actually help with that. We're using AI to design more efficient base editors, prime editors, gene editors. You're using AI to design the mRNA molecule that expresses those molecules in your cell so that the first design you go with actually works. When in reality, a lot of times companies can get tripped up for many years trying to design an mRNA molecule that actually expresses well. So I think that's an exciting direction, and also just using AI to simulate specificity and function in cells, right? Like there's a future where we have virtual cells, virtual tissues, virtual people, where maybe you could even just simulate millions of people in a phase 1 trial or a phase 2 trial, right? And I don't think it's inconceivable, maybe this is decades away, that you could go straight from simulation to maybe a phase 4 or a limited phase 4, right? Where that could just drop many years off these timelines. And so that's a really big vision thing, but that's where I think the prospect for AI in biology and medicine should be headed. So.
Great. So yeah, I think obviously lots of really exciting things looking over the horizon. Maybe the take-home message here, and as David nicely articulated in our prep call, is that the science really is not limiting anymore. It's part of the process in the system. And as we have innovation on all aspects of this, we can start to think about getting treatments into patients much quicker. So please join me in thanking the panel for this exciting discussion.
Thank you.